Good afternoon and hi everyone. So welcome to the 2024 edition of Liver Seminars. So today I have the pleasure to introduce the first speaker of the year, who is other else than Theodore Alexandrov. So Theodore is a pioneer in the field of single-cell metabolomics. He is team leader at the MBL, head of the metabolomics core facility, and also a faculty of the Molecular Medicine Partnership Unit between MBL and Heidelberg University. And he's also a principal investigator at BioStudio and the Bioinnovation Institute in Copenhagen. So the team of Alexandrov at MBL aims to reveal the secrets of metabolism in time and space, in tissues and also in single cells. They have several experimental and computational technologies, softwares, and resources that we are going to learn today about. So he has like more than 100 publications in top journals, multiple patents, and a lot of startups. And one thing that is very interesting, and I hope that he will comment, is Metaspace, which is a cloud-like open source that is very interesting if you are interested in spatial metabolomics. So I don't want to take more time from him, so I would just let you begin.
Thank you, Douglas, very much for the introduction, and thank you and all organizers for inviting me to speak here. It is very, very exciting to connect with in general and community and present some of the work that we are doing. So today I'll present you technology and actually a set of technologies or set of tools in the field of spatial and or single cell metabolomics. And I'll focus specifically how can we use this data to look at the concept of metabolic cell state. And my name is Theodore, and I work at European Molecular Biology Laboratory, which is an amazing organization, intergovernmental organization in Europe with several sites. I'm located at MBL Heidelberg in Germany, but there is also multiple sites. And it's such an amazing place to work, and I'm so fortunate to lead a team there. But I also recently we started commercializing some of the technologies, in particular those presented here at the Bioinnovation Institute in Copenhagen. And first, I would like to introduce our team in Heidelberg. And here you see each of them. And this were some fun activity where we presented these individual Pokemon style cards for everyone, and they picked their strengths and weaknesses. And I'm showing it here because to demonstrate the diversity of the backgrounds, because here with the color, they indicated whether they have background in biology or mass spectrometry or bioinformatics, data science, or software development. And it's so cool to actually work and lead this very talented group of people who combine expertise coming from different angles. And quite often from biology, we formulate the questions that we develop methods for. And as you will see, we use mass spectrometry, and we benefit here so much from devoted scientists and experts working in it. A lot of data generated, that's why we need bioinformatics. And we and others benefit a lot from having devoted software engineers. And it's also very cool to actually work in different environments. And here I'm showing our commercialization team in Copenhagen at the Bioinnovation Institute. And you see it's completely different vibe, a different style. And here we're trying to commercialize, we're working on commercializing single cell metabolomics, in particular for drug discovery and personalized medicine applications. But before I'll jump into single cell metabolomics and spatial metabolomics, I would like to explain why are we doing all this work. And this why probably will start like, why metabolomics? Because, I mean, for some people, it's a question like, why omics? But you probably heard about omics. You probably heard about genomics, of course, like transcriptomics, proteomics. Metabolomics is still new kid on the block. And quite often it is being discussed as metabolites that are produced at the end of this flow of information through the central dogma. And these are metabolites which are readouts closest to the phenotype and function. So, and this is certainly true, but what is very, very interesting, and the attitude towards metabolites was changing over the past decade. If I had to choose one publication to highlight this change, it probably would be this one, a short opinion piece, very easy to read. Check it out. In 2012, about 11 years ago, so they postulated that metabolism is actually not one-way street from DNA to metabolites and function, but it's actually two-way street. And there is this reciprocal regulation of metabolism and signaling. And what they mean here is that metabolites, they actually was revealed. They play different regulatory roles, controlling molecular machines at the higher levels of the central dogma through epigenetic regulation, chromatin modification, RNA modification, and of course, post-translational modification, which is probably one of the most known. So, and this, like over the past decade, kind of changed the perspective and the view on metabolism and metabolites. They're not just there's some end products, but they actually active players of the cellular programs. And now there is even more revealed, but also new discoveries are coming that about the function of metabolites, how it controls either DNA, RNA, or other molecular machines, or reprogramming through 3D organization, through molecular switches, and through providing molecular microenvironment, affecting, for example, protein folding, and so on and so forth. So it was very interesting to witness over the past decade how this view on metabolites was changing, that they are not just building blocks for cells like amino acids and lipids for membranes and so on and so forth, or fuel producing ATP, but they're actually active players of cellular programs. I think this was fascinating. This also was changing the attention and interest in metabolism, but also metabolomics. But till recently, it was very hard to detect those metabolites. This was changing. And not only it was changing for so-called bulk metabolomics, but also people started asking questions about spatial or single-cell metabolomics. And here I would like to focus on our motivation for spatial and single-cell metabolomics, because what we do in our team, we develop methods which can do spatial single-cell analysis and detect metabolites. So, and the two major motivational topics here, one would be tumor microenvironment. And here we can, in particular through the efforts of the past decade, we know about heterogeneity, single cell heterogeneity here and spatial heterogeneity. There are so many different cell types here, tissue-associated macrophages, tumor-associated macrophages, and there are cancer cells, adipocytes. Of course, in liver, this will be different, but there is of course, hepatocytes. There is multitude of immune cells, like Kupffer cells, and also T cells. And so there is multitude of cell types. But what was even more interesting that was discovered also through single-cell RNA sequencing, that the cells actually reside in different states. For liver, this is in particular interesting and important because the same hepatocytes as cell type, they actually they reside in different states due to zonation, right? And there is many even more states, and some of them are metabolic cell states, which differ, not surprisingly, because in particular solid tumor microenvironment, it has these gradients of pH through lactate secretion by cancer cells, also hypoxia through the oxygen overconsumption, and all aspects of metabolic waste. So we're dealing here with heterogeneity, with metabolic gradients and metabolic microenvironment. And of course, here we need the technologies that will be able to dissect all this. And the second big topic where spatial single-cell metabolomics can be very helpful is immuno-oncology. And here I'm showing a review from Erica Pierce and colleagues, where they discuss immune cells and they compare them to nomads, which are born in one place, they get educated in another place, and now attracted by chemoattractants, they actually get attracted here, they squeeze through the blood vessel wall, and now they need to adapt immediately to a very foreign environment. So, and here they would face all the same challenges that cancer cells have here in terms of what we have discussed here, metabolic, not heterogeneity, but acidity, like a lack of oxygen. They need to compete with the cancer cells for nutrients, and they also can go in exhaustion. And now there is more information that functional exhaustion of T cells is actually linked to metabolism. And of course, here is very, very interesting and has high potential to do metabolically optimized bioengineering T cells, such as metabolic priming of CAR T cells. So here, of course, there is a lot of interest and demand for having technologies that will be able to pinpoint what is the metabolic profile or what is the metabolic state of this cell or this group of cells in that or another specific condition. So ultimately, we know now there is a lot of evidence that metabolic state is linked to cell function. This is an abstraction which can help us interpret metabolic levels. Now the question is like, how can we detect metabolic states of immune cells, cancer cells, but also microenvironment? And this is where what our team is mainly focusing on is develop methods specifically for this, for spatial single-cell metabolomics, to provide context, in particular spatial context, dissect single-cell heterogeneity, provide readouts which are closest to the phenotype, but also linking it to the metabolic cell state. And this is pretty challenging because metabolic cell state is a very dynamic phenomenon. Even in terms of sample preparation, we need to design experiments and optimize them in such a way that we can preserve this metabolic cell state. But of course, there is even more challenges when we start interpreting this in contextual biology. So how do we do it? We use mass spectrometry as the main readout for levels of metabolites within the cells or tissues. We motivate pretty much all developments we do by questions from biology. And we use more and more AI and ML, artificial intelligence and machine learning and software to be able to really dissect all this data, make sense out of this, and interpret this. Now I'd like to show you how this technology can look like. So here you see multi-imaging mass spectrometry, which is the main type of mass spectrometry which we use and others for spatial metabolomics. So MALDI here stands for matrix-assisted laser desorption ionization. So it is a laser-equipped mass spectrometer. So here you can place a pretty much regular histological glass slide with a tissue, and here is actually a liver tissue section on it. You can put it inside, you can close the lid, and there will be analysis performed. But imaging mass spectrometry comes in different flavors, and multi-imaging mass spectrometry also comes in different flavors. Here I'm showing you our setup in Copenhagen. You see it can hold like 2 slides, but what's even more important, for cells, we can put a lot of samples here, and this mass spectrometer is 20 times faster than what we have in Heidelberg. So we have actual potential to perform much faster analysis. But the principle is still the same. So what happens once we put this, for example, this liver tissue section on a glass slide, the laser will be firing a particular location focused at a very small area, 5 to 10 micrometer pixel size. And then the molecules, there will be a lot of energy focus there. The molecules will be, it will be sort of micro explosion happening. There will be plume of molecules shooting in the air. And this plume will get captured by a mass spectrometer through vacuum, will get captured inside, molecules will get analyzed, will produce this mass spectrum, which is sort of quantified barcode of which molecules are at this pixel and how many of them are relative to amount of other molecules. So then we can shift the stage, we again shoot laser, capture, mass spectrometer, shift, and so on and so forth. In a pixelated grid manner, we can collect information from more than 10,000 or even 100,000 pixels. So, and once we did this, we have a lot of spectra, but we can also select a particular dimension of this spectra corresponding to a specific molecular weight of a molecule. And now we can map intensities of all the peaks onto where they were sampled from. And now what we have an image which represents the relative abundances of a molecule of interest in our tissue section. And then through the process of metabolite identification, we can figure out which molecules are actually hidden in all this data set. And it's very important because if we look at the properties of this technology, it can perform analysis with 5 to 10 micrometer pixel size. Once again, it's pretty close to single cell resolution. We can also make the pixels bigger if necessary, but this is like the best one can get on commercial simulation. We can detect more than 100 molecules in one shot. Very important can be untargeted detection of metabolites, small molecules, amino acids, glutamine, glutamate, and many other glycolysis, TCA, and urea cycle. Urea cycle for liver is very important. So, and lipids, fatty acids, and drugs and drug metabolites. And since technologies generates such long spectra, it generates a lot of data. So this is where we are coming from. Our background was originally computational, but now we're developing methods at the intersection of experimental and computational analysis. What's very important is that this technology, specifically imaging and specifically multi-imaging, became the major technology for spatial single-cell metabolomics. If you're interested in this perspective, you can check out this review where I was discussing this and where this field stands and so on and so forth. So, and Douglas mentioned that Metaspace. Metaspace is our software actually to perform exactly this step. So for metabolite identification, I actually was not going to show it today because I was planning to show more cell analysis and more biology-specific analysis. But if you are someone who has already imaging mass spectrometer, in particular multi-imaging mass spectrometer, and you're interested in metabolite identification, definitely check it out. Metaspace, I think I mentioned also here in this publication, this is a free and open source platform for metabolite identification for imaging mass spectrometry. Lots of, we have more than 2,000 users. We have a lot already publications of our users, so exciting. And also many public data sets that you can compare. And there will be a few very exciting publications also coming out on this soon, how to do it better and how actually extract biological information out of those public data sets. But this will not be covered today. What will be covered is actually how can we use multi-imaging mass spectrometry to perform single-cell metabolomics? Because you probably, all of you are in some sort of biology or related to biology field, and you know the single-cell omics, in particular single-cell transcriptomics and RNA-seq, were completely transformational for biology. We now live in pretty much era of single-cell biology. Since 2015 or 17, this changed the way how we do analysis, molecular analysis biology. So what about single-cell metabolomics? Single-cell metabolomics probably all of you or many of you haven't heard of because it is an emerging field. But there is a lot of attention, there is a lot of interest, and there is a lot of developments. There is so much going on I will not be able to cover, but I want to just bring two highlights here. So in 2021, about 2.5 years ago, single-cell metabolomics was recognized among top 10 emerging technologies in chemistry by IUPAC, which is the main chemistry standards institute. And last year, single-cell metabolomics was recognized among 7 technologies to watch by Nature. And this is aside such major achievements of humanity like James Webb Telescope and so on. So there is a lot of interest. By the way, this year, there's also single-cell analysis in 7 technologies to watch, but it's focused more on cell atlases. Check it out. I think that the paper was just out. So, okay, single-cell metabolomics, a lot of interest. So how are we contributing here? So we are contributing with the method SPACE-M and many other developments around this. And SPACE-M stands for Spatial Single-Cell Metabolomics. The method was developed by our first PhD student, Luca Rapes, and we published it in 2021, specifically with application in liver. So, and how the method works, we take cells which should be either cultured or plated on glass. Then afterwards, we do microscopy to really find where the cells are, to segment them individually, also to find their phenotypes in terms of morphometric phenotypes, but also in terms of their fluorescent phenotypes, because we can combine it with a small molecule staining, but also with the fluorescent reporters. And we're now even developing the method how to combine it with the immunofluorescence. So then we perform multi-imaging mass spectrometry introduced earlier, and we get these pixelated images, but even at the resolution of 10 micrometer pixel size, then it's still not single cell information because one cell can produce multiple pixels. And also one pixel can correspond to 2 cells. Then we run this data through our platform Metaspace, which again, it is for tissues, but it is actually just for imaging mass spectrometry. So we find which molecules are hidden behind these images. And then Luca has developed how to overlay computationally these two types of data, microscopy and image mass spec, with a subcellular precision, and also how to deconvolve the contributions of one cell to pixels. And now after this deconvolution, which is done with a mathematical formula of normalization, we can assign back intensities to cells, where they're coming from. And now we have truly single cell metabolite images, where for every cell we have an intensity, relative intensity compared to other cells. Or we can flip this data matrix, and now for every cell, we'll have relative intensities of metabolites or lipids or drug metabolites, small molecules, detected there by mass spectrometry and identified by metaspace. And also we have information about staining, morphometric properties, spatial features, and actually just images, microscopy images of individual cells. And the technology can now perform analysis detecting more than 100 metabolites from more than 10,000 cells per hour. This is possible only with a high-end, high-speed mass spectrometer that we have in Copenhagen. But with this one, it's very, very fast. And we can also analyze pretty much almost any cell type. Now I'd like to show you our study that we have performed in the context of non-alcoholic steatohepatitis. This is a bit of old study for us. Again, it was published 21, but I would like to show it simply because I think it is probably the most relevant work, published work, that we can show now. There are some follow-ups, and there is one very interesting, also NASH follow-up, but we haven't published it yet. So I'll show you what we published in 2021. So we consider it here non-alcoholic steatohepatitis, which is common factor for liver cancer. We know that it is in particular influenced by excess of free fatty acids, pushing the liver towards accumulation of free fatty acids and lipids, which triggers inflammatory stage of fatty liver disease, which is called NASH, which is a main factor towards fibrosis and HSE. So in particular, because it's actually triggered here by cytokines created by immune cells, in particular IL-17A, IL-17A, and others as well. So here we teamed up with Matthias Heichenwälder, who works on liver. Probably many of you know him in the context of also immuno-oncology, mouse models. And they, in particular Mira Stadler, from PhD, back then PhD student, they had a beautiful in vitro model of NASH, which is differentiated human hepatocytes, HepRG, supplemented with free fatty acids, in particular oleic and palmitic, and inflammatory factors like TNF-alpha. So in here, if we stain the cells with the lipophilic staining here shown in yellow, Hust shown here in blue, nuclei, we see that a lot of cells, they show this pronounced steatotic phenotype, accumulating lipid droplets and really bloating with lipid droplets. Interestingly enough, then even in this isogenic population of cells, some cells do not show this phenotype, even if they're neighboring to each other. So there is a pronounced single cell heterogeneity. So we worked together with Matthias, Mira, and others. And first we considered, can we characterize static state of hepatocytes? We took them, the cells stimulated with just fatty acids. We stained, we see that there is accumulation of lipid droplets, but the question is like, what are the lipids accumulated there? We performed our analysis. I'm showing you UMAP, which is analysis similar to PCA or principal component analysis, but is a bit more sophisticated and the standard way of visualizing data, single cell data. And now if we see that the cells organize themselves in two subpopulations, particularly this dense subpopulation, and now we can get microscopy readout from this data, which is a unique property of this technology. We can map the fluorescent readouts showing lipid droplet accumulation to the cells, and we see that those cells actually have a lot of lipid droplets. And now we can find markers, in this case, this particular lipid called triglyceride 48-0, having high levels in these cells. There are also some markers which have low levels. So we performed statistical analysis to really characterize molecularly the state. We found the correlation, and this is really cool because we did correlation here between molecules and fluorescence. We found the molecules which are most correlated here with fluorescence. We did enrichment analysis using lipid ontology developed by Marta and Molenauer. We found that it's mainly triglycerides. It's the lipids which are normally associated with lipid storage, lipid droplet. So this was very good. We found some markers, but of course we were interested to do more advanced analysis, in particular analyze models of NASH, where we have not only fatty acids, that would be more in vitro model of NAFLD, but pro-inflammatory cytokines. And we also here, we inhibit an inflammation with TPCR1, which is a small molecule inhibitor of NF-kappa B. And phenotypically, we see a lot of difference. There is a lipid drop accumulation, there is drop here. But again, the question like, what is going on molecular level? We gain performance-based M. I'm showing you results for 27,000 cells, 700 metabolites and lipids per cell. Cells organized here in these subpopulations actually separated a lot by the conditions. Here, unstimulated, this just fatty acids plus IL-17A, and then treated with a small molecule. So, and we see that they shift upon the stimulations. But what's even more interesting, if we cluster the cells here, then we see that here there is quite strong heterogeneity because we have not only these cells, but we also have yellow and blue cells. The blue cells reside in a steatotic state. They are non-responders to our treatment. The yellow cells here, they're also actually molecularly very interesting because they demonstrate that metabolically, they reprogram themselves to reside already in steatotic state, although they didn't experience the stimulatory effect of cytokines yet. It is a small percentage of cells, but we show that just fatty acids is enough to reprogram cells to go towards steatosis. We validated these results by finding pseudo-time trajectory, which is associated with inflammation, finding markers which are expressed at the end of this trajectory, and then comparing the intensity of these markers in cells with in vivo analysis, where we took mouse models of NASH, early NASH, normal diet versus Western diet, and we considered only livers of these mice, and we performed analysis, a bulk, a lipidomics analysis, and you see how well the data found in cells actually is corroborated with the data found in vivo in mice. So, and also very interestingly, several markers here and several markers here, they were reported actually in the context of human cohorts and NASH studies. So this was very, very exciting. Again, this is where we were in about 2021, already contributing to single-cell metabolomics and really seeing how it is emerging as a new field. So now we have a method for spatial single-cell metabolomics. And back then already we demonstrated that it reveals metabolic states in steatosis and in NASH. And now we have actually new results that I'll not show them today. I'll show you some other study. We can detect not only lipids, but we can detect metabolites from different pathways like glycolysis, TCA, pentose phosphate pathways, amino acids, glutaminolysis, urea cycle, and so on and so forth. And on top of this, we actually have now even higher throughput methods. We can analyze even more cells and samples and even profile very, very challenging, but we finally can profile metabolic reprogramming of T cells, which are not even adherent cells. But today I would like to show you some other study, which is first, I think, glimpse, at least for me, that single-cell metabolomics and this sort of analysis can be relevant in patient samples. And I'll show you one study, which is, at this point, is not related to liver. It is more technology showcase. But again, I will focus on one aspect I like later, which I think is very, very exciting. And also very exciting, not only in the context of PDAC or pancreatic cancer. So the study is specifically on single-cell lipidomics. And here we were discerning phenotypic difference in pancreatic ductal adenocarcinoma, which is type of pancreatic cancer. This was amazing work by our mass spectrometrist and computer scientist Gianni, and the experiments were performed by our lab coordinator Shoraz. And here we teamed up with a team of Jens Sivike from University of Medicine, University Hospital Essen in Germany. We focused on PDAC. This is a very common and actually aggressive cancer. I'm pretty sure that some of you had some maybe relatives who got affected, or you've heard stories that the survival of this cancer is very, very short. So, and there is a specific subtype or phenotype of PDAC, which is even more aggressive, which is this metastatic. And in this case, for metastatic or aggressive type of PDAC, the 5-year survival is 3%. So here we were teaming up with Jens and particularly Maria, who is a senior scientist on the team, to really characterize molecularly epithelial versus mesenchymal. And mesenchymal phenotype is that phenotype which actually leads to metastasis and high aggressiveness. And for this, we use single-cell lipidomics. So the study we performed was the following, and I think this is, for us at least, it's a really unique aspect of the study. So we analyzed here cells coming from very different sources. So we analyzed cell lines, and for us, this were prototypical differences between epithelial versus mesenchymal. But of course, cell lines reflect only to the limited level the differences and molecular programs and reprogramming that we observe in patients. So here, Maria and Jens, they actually have a lot of expertise on how to prepare cells in very different ways. For example, they take tumors, they graft them, and afterwards they take the PDXs and then they plant them back and passage them to actually derive patient-derived lines. They also have primary tumor cells, and this will be coming just directly out of the solid tumor. They also culture patient-derived organoids. And then for those organoids, we can also plate them back on the slide. And also they captured here primary malignant ascites from the body fluids from the patients. So we had the whole array of cells, but coming from 2 groups of patients, either epithelial or highly aggressive mesenchymal phenotype. So we plated them on a glass slide. Tremendous work by Maria. Then we did mass spectrometry analysis, again, SPACE-M, as we normally do. And I'm showing you the results, which are unpublished, but I think very, very exciting. So we have first focused just on cell lines. Once again, cell line, this is just ATCC cell lines. Some of them, they're non-epithelial, non-mesenchymal, either based on protein markers or transcripts. And we just run all of them in untargeted fashion through single-cell lipidomics using SPACE-M. Then we see that they organize in these 2 lobes on your map. And if we started actually mapping back the phenotype, epithelial versus mesenchymal, we see that this is the phenotype which drives completely unsupervised fashion, drives the difference between the cell lines. I think this was very exciting already. So we looked into the markers which separate all epithelial versus all mesenchymal cells. We found the markers, similarly how we did in the liver analysis, which are expressed here and there. And they were matching actually what we know that from epithelial to mesenchymal shift through, for example, EMT. So there is decrease of ceramides, and this is a special class of lipids. So there is altered sphingolipids composition and this specific class. Okay, this was already very interesting. We performed enrichment analysis using finding the other classes of lipids, which specific, like yes, ceramides. And we also know that in mesenchymal, for example, the function of these lipids is increased lateral diffusion. So it's all kind of matching. So, but also some other classes of lipids. So in particular, what was very interesting that in mesenchymal, we actually saw accumulation of triglycerides and molecules involved in lipid droplets, similar how we saw it actually in NASH. So we validated this again by doing staining, lipophilic staining, oil red here, and we indeed saw more lipid droplets accumulate in the cells, in the cell lines, which are known models for mesenchymal cell. Okay, now what about patient data? So first, we actually took patient-derived xenograft cells, because remember we had different sources there, but xenografts, we took them as the first one, also because we had most patients with this. And if we now do actually a very similar analysis, we take only PDX cells, we do a single-cell lipidomics, we do UMAP, then we again see very similarly that cells coming from different patients, and now patients were characterized based on facts, markers, and transcripts, were characterized either epithelial or mesenchymal. So we see that there is indeed the difference, but this difference is actually more fuzzy, which is not surprising because these are primary patient cells. So, and we see similar story again with different groups of lipids. And again, for mesenchymal, there is above lateral diffusion. Okay, this makes sense. And specific classes that we found also that were reported in the context of epithelial cells, in context of EMT, in another type of cancer. And now the last step, what we did is we took all the cell types together. So we took our cell lines and we took, at this point, we have 10 patient-derived samples. We're increasing this number. 5 of them were PDX, 2 of them were PDOs, patient-derived organoids. 1 was primary tumor, and then 2 were coming from ascites. And altogether, after we performed this analysis, we had tremendous amount of cells. We had more than 100, almost 150,000 cells here. And if we now again do a UMAP analysis from all this almost 150,000 cells, and we first map which of them were characterized initially as epithelial, either cell lines or like patient data, we see exactly the same story. Mesenchymal phenotype on one side, epithelial on another side. And what's interesting that we had, I think for 2 patients, phenotypes which were characterized as intermediate, and they were like localized here right in the middle of this UMAP. If we now look into specific cell types where they're coming from, then we'll see that bright green is actually cell lines. And cell lines, they are the most different. You see like PDXs, PDOs, they are bits here in the middle. Cell lines, they're the most different here in mesenchymal and epithelial. Not surprising because again, they are models. They're models for our phenotype. They're well characterized. They're most pronounced. And this is also what we see here in metabolomics. I think it's also very exciting. What's even also what this UMAP shows, and I'll not go into details here, that but it's very well visualized by this UMAP and target analysis. We have exactly the same lipid markers through cell lines, PDXs, PDOs, and tumor cells. And I think this is very, very exciting. And let me pause here to highlight it. So what it means is that if we want to study metabolism of a patient, for example, in context of a tumor, then the cells that will prepare from the patient for this molecular analysis, the metabolism and metabolic profiles and states of the cells will be preserved through our cell preparation, even if we go for PDXs. Which, I mean, think about this. We graft PDX into mouse, we leave it proliferate there, then afterwards we sample it, we plate it again on a glass slide. One can imagine that, and because metabolism is so plastic, so dynamic, but it's actually apparently so tightly regulated that there is a metabolic memory of the cells concerning this specific phenotype all the way back to the patient. And I think this is very, very exciting because it shows the potential of using patient-derived cells for molecular phenotyping and also for other molecular analysis. I think this is, again, very, very exciting for us. We were so happy to see this. There is more coming here. We train the predictors. Yes, we can predict the patients based on their profiles. And we also do molecular characterization, hoping to submit this study very soon. So here I would like to highlight that, yes, single-cell lipidomics that we previously have shown in the context of NASH and liver, it can do more. And also it predicts, for example, in this context, PDAC, patient phenotype. I think it's so exciting that markers also in line with literally lipid functions. And what's, I think, the most, for me, exciting aspect of the study for us, but I think also for anyone, that the markers and profiles, they consisted across multiple cell types. That means those cells, they actually have metabolic memory of phenotype they are coming from the patient, and we can use those cells for advanced assays. So let me check how I'm doing with time. So I think I'll not include any other studies, but I will take time to acknowledge everyone here And this is our lab coordinator, Sharaz, our mass spectrometrist, Mons. Louisa, I didn't highlight her work. She works on immune cells. I really wanted to show it, but I'll do it at some point later. So Gianni, she's an amazing scientist doing this PDAC project, and many others. So Luca was a first PhD student in our team who really absolutely kicked off this line of work by developing SpaceM, and many collaborators from the GFZ, this actually, I didn't show this study, and University Hospital Essen, of course, Jens and Maria, some companies with whom we are working, all the funders that we perform this work. And thank you so much for your attention. And also, just in case you have any ideas for collaborations or anything, we actually will be moving to University of California, San Diego this year. I'm so excited to move there, and many members of our team will also move. We'll be in Department of Pharmacology, pushing even more the single cell and spatial metabolomics developments, computational experimental method development, but also with actually focusing more in particular on clinical and preclinical applications. Thank you so much for your attention. I'll be so happy to answer questions. I think we have time for this. So the main reference method were immunofluorescence, where we use known markers of epithelial versus mesenchymal. So since this was performed, we also have collected, double check to validate all this. We collected bulk RNA-seq to also use gene expression, also the known markers of epithelial versus mesenchymal. And on top of this, since then, because the cells were sampled a few years ago, we actually have clinical follow-up information about aggressiveness. And indeed, for many patients who were characterized as mesenchymal. So yes, so we also have the clinical history for this as well. So yes, absolutely. There is a lot of interest to integrate single cell metabolomics with other readouts. And what we are working on right now is to integrate it with multiplex protein profiling, so that we can characterize cell types and we can characterize some basic phenotypes like proliferating with Ki67 and so on. So this is possible, but we still need to optimize a few parameters. So what was also shown that, and how is it possible? Just that we first need to still do single cell metabolomics, because metabolites, in particular small molecules, they're prone to leakage, they're prone to transformations and so on. If you do any sort of immunofluorescence or any, for example, fish fluorescence in situ hybridization, you will be losing those metabolites. So that's why we first do single cell metabolomics. But very interestingly, we can tune mass spec to such a way that despite this process, I told you that there is this micro explosion happening and there is material loss. We see, and it's also shown more and more, that after imaging mass spectrometry, there is enough material, and in particular like macromolecules such as proteins and also RNA, mRNA left, so that we can use immunohistochemistry, we can use immunofluorescence, immunohistochemistry, we can also use fluorescent in situ hybridization. It was also recently shown in Nature Biotechnology that when you do it for tissues, not single cell, but imaging mass spectrometry can first detect metabolites, and then afterwards you can do spatial transcriptomics, where the technology is like Visium. So it is possible to combine it. On the single cell level, it is still pretty challenging, but it's possible. Now concerning fresh samples. Yes, so I'll answer first concerning cells, that for cells and tissue situation is slightly different, because cells can be prepared in the conditions that not to perturb the metabolism, to keep metabolites inside. And then what we do before the analysis, similar to any imaging mass spectrometry, we desiccate them. And once the cells are on the glass slide and they are desiccated, they can be shipped, they can be stored at least for some time. So we evaluated at least for a few weeks. Yes, we can do this. And this is how we work, for example, with collaborators. Ideally, they prepare cells, they plate them, they desiccate this, they ship to us on dry ice. We get them. So, and now concerning tissue samples and in general, what Sergey brought up. Yes, and also what Douglas highlighted. It is, for metabolomics, is so important to really trace the life of your sample before it gets either frozen or metabolites extracted, or like cells or tissues are desiccated, because there is so many things where you can cause the degradation, for example, in animal, or there can be degradation also non-enzymatic, like oxidation. There can be also some additional factors, leakage of metabolites, and so on and so forth. So which are very important. So for tissues, the normal protocol is flash freezing. And this can be applied either to tissue to animal models where organ you extract it as fast as possible. There is some degradation happening. Metabolites with a very fast turnover, they will probably be, their levels will change. But many metabolites, they don't have such fast turnover. If you extract it within, say, minutes to half an hour, then you flash freeze your organ. At this point, metabolism is very, very, very slow, almost stopped. So, and you will observe a difference if you store it for like weeks and months in the years. But you flash freeze, then afterwards you do section at -20, and then you immediately desiccate, which is a very fast process because the tissue section is 10 micrometer thickness. So this is normal procedure, but this requires frozen material for imaging mass spectrometry. So unfortunately, all the biobanks with FFP tissues, they cannot be used for this. But on the upside, now with all spatial omics, spatial transcriptomics, and so on, they also require frozen material, and there is more and more availability of this material. So, yeah. So this is, I think I covered both questions. We don't have the results on this for single cell analysis yet. We do have results. We have a project that I didn't show today, where we considered a very simple tissue models, and we performed for 2 tissue sections. Here we performed spatial metabolomics, not single cell, but near single cell. And for this consecutive section, we performed spatial transcriptomics, very high subcellular detection, multiplex FISH principle for 100 genes. Half of them are cell type markers, half of them are metabolic genes. And it was very, very interesting because what we see that for some cell types, we have one-to-one. We have one-to-one correspondence between cell type and what we call, it's either can be called metabolic state or metabolic phenotype. And so we have one-to-one, but for some we have actually, for the same cell type, which we have 2 metabolic states discovered in data. And for others, we have for 2 cell types, we have 1 metabolic state. So some of these results potentially can be due to inability to resolve cell types, because if they are really peppered within the tissue, like we consider it brain, and in brain astrocytes, they're really scattered everywhere. So it's very hard to resolve them. So this may be just limitation of technology. But we see an indication that what we're detecting also that one cell type can be in 2 different metabolic states, and that's why we see like this in the data. So this is preliminary results, but we're already performing this again at near single cell resolution. The single cell, we need a bit more time. I'll touch first the slides. So in general, we use normal slides. Sometimes we code them with either PLL or PDL, and we see better adherence, but this is pretty common. So we experimented with a few other slides, but we actually found that this is not necessary. So it is not actually the slides, but it's more like culturing conditions, because some cells, for example, they don't like to be cultured in small wells, and so on and so forth. So, and So this is mainly a bit of a problem. And the biggest problem we faced, of course, for suspension cells, like T cells. This was the most challenging, but still, even for T cells, we actually use regular slides. The tricks were in really fine-tuning the culturing conditions and deposition conditions as well. The T cells turned out to be very, very sensitive, and the adherence like properties and the quality changes a lot depending on culturing parameters. Now concerning the metrics, this is about metrics, which is like small molecule, which enhances MALDI imaging. So actually for lipids, most of the time we use the standard ones. And we'll be publishing actually studies soon showing that standard matrices, they work very, very well. And it's comprehensive inter-lab study. So normally for lipids, we use DHB, and for small molecules, we use DAN. So we're missing a little bit amino acids. When we want to detect amino acids, then we use 9AA. No, I think that for amino acids, I think also use DHB. Sometimes we use 9AA to have better detection of some molecules. But again, these are all standard matrices. So actually, this was not tumor microenvironment. So we started with a much simpler model because we also wanted to validate this method and have confidence. So we worked with cells from peripheral blood and isolated. We worked mainly with CD4 T cells, although we actually tried some other cell subtypes. And here we focus mainly on phenomenon of activation. And also we sort of performed analysis, which is normally called seahorse analysis, where you push cells into different states, metabolic states, inhibited glycolysis, inhibited respiration, and you really test how the profiles change. What we have found there is the following. First of all, we found that cells, upon inhibition of glycolysis, they actually use a compensatory mechanism, which is likely pentose phosphate pathway. Second, we also have found that there is very interesting heterogeneity of CD4 T cells in the activated state. So they're not homogeneous at all, in vitro metabolically, not like even close to what we saw in single cell lipidomics. I think this is very, very interesting. We're still validating this, and but there is like very consistent different clusters of cells. And the last thing that we have found there, which is also, I think, very interesting, that when we do activation of the cells, I think we use like standards CD3 and C28, I think, beads coated with these markers. Then when we first check the activation with the markers like CD25, CD69, which are standard markers for early late activation, we see that about 70 to 80% of cells, they express those markers, which is very much in line with the literature and our collaborators and so on. But when we do single cell metabolomics, we see that 97% of cells, they completely shift their metabolic profile into activated metabolic state. And I think it's very interesting discrepancy that in terms of expression of the surface markers, we see 70 to 80%. Okay, measure it with antibodies, which are probably binding to specific conformation of these markers and so on. Again, here, we're really not experts, but this would be an assumption, right? So whereas metabolically, we see all of them are changing. So that means for us, it's kind of also a very interesting result because it shows that, for example, for this phenomenon, single cell metabolomics is very sensitive and reflective of the functional change that we induce in the cells. So these were a few takeaway moments. Yeah, I think it's probably concerning that thing that I mentioned with patient models, not patient, but patient cells and particular PDXs. I'm coming not from biology, not from clinical cell biology. So for me, this was very surprising. I started researching. And because for me, it's like, how can this happen? Just imagine what kind of perturbations cells go through in PDXs. So, but there is actually some body of literature where the same phenomenon was observed for other omics, not for metabolism and not for metabolomics, but for example, for transcript profiles, transcriptomics. So I think it's very interesting. I read already a few papers where at least not mechanisms, but there's some discussions about this. So it definitely was observed. And I think it's really, really cool. Shows probably the relevance of PDXs in general, in particular for some types of analysis. And so I think it's, but what are the mechanisms? Hopefully someone will discover and tell us.
If there's not any more questions, I would say that maybe it's time to close this seminar session. Mm-hmm. So thanks once again for coming.
Yeah, thank you, Douglas, and all organizers. Thank you very much to the participants for Q&A and active participation. This was very interesting to read your questions and where you're coming from. And if you have any ideas, any questions, do not hesitate to reach out for collaborations, but also if you have any questions concerning imaging mass spectrometry, single cell metabolomics, how it can be used, where it can be used. So please do not hesitate to reach out. And once again, it was my pleasure and looking forward to seeing you in some other setting here.
Okay. Okay.
Mm-hmm.
So,
right. Cheers. Bye, everyone.